Dr. Rizzoti, thank you very, very much indeed for coming to spend a couple of minutes with us at eCancer TV in Liege at the ENEA meeting. You work at Mill Hill, you're from France.
Yes.
And you're working on embryonic stem cells in the pituitary.
Yes, exactly. So we have been, I am a developmental biologist, and we have started to look at the development of the pituitary and the embryo and characterize this expression of SOX2, which is a protein associated with stem cell in different systems, and that has recently been shown to be necessary to reprogram somatic cells into induced pluripotent stem cell. And we follow its expression in the embryonic pituitary, and we realized that it was also expressed in the adult pituitary. And based on this expression profile, we have been able to characterize the cell population in the adult gland that we think represent adult progenitors.
And this is in human tissue?
This is in murine tissues for now.
In murine tissues?
But
okay.
The expression is maintained also in humans.
In humans, okay. Why the pituitary?
Well, the pituitary, because we had a specific interest in this gland for historical reason, because we've been working on SOX3, which is also involved in hypothalamic pituitary axis function, and because I think the system is, of course, very important physiologically and also experimentally quite accessible, both in the embryo and in the adult gland. And moreover, we know quite a lot about the differentiation of the different endocrine cells in the pituitary. So it's a good system to look for the involvement of a putative population of progenitors.
And how do you know the progenitors?
Well,
this is always a problem.
Yes, exactly. We think we are really confident they are progenitors because of all the experiments we have done in vitro now. So when we select for the SOX2 positive cells from adult pituitaries, we are able to form spheres in cultures, and initially these spheres do not express any hormonal markers, or they are undifferentiated, and upon modification of the culture condition we are able to obtain endocrine cells from these SOX2 positive cells. So this is a very good argument to say that they are progenitors.
You have an in vivo model?
So we are working toward an in vivo model now. We are creating genetic tools in the mouse to go further and to be really able to prove this hypothesis formally.
And what are you going to do with the results once you show that this is the progenitor and that's the pathway? What are you going to do?
So what I really would be interested in, adult stem cells are really important nowadays, and everyone looks in his favorite organ if there are some stem cells maintained. And what I'm really interested in is to find in the normal physiology of the organism, when do you use these stem cells, what are the modality, and how often are they used, and also in pathological condition, I mean especially of course cancers, what is our involvement, and this is really what interests me.
And have you the possibility of getting the genetic signature of these cells once you've proven what they are?
Yeah. So by now the genetic signature is really simple. So we think, so other groups are working on these cells, and they have added new markers of them, and we think that in the future the list of markers for these stem cells will be longer and will allow us to be a bit more precise, because by now the population we are working on is clearly heterogeneous. So we think with more markers we'll be able to really come down to the true stem cell.
Dr. Rizzoti, thank you very much indeed. Really appreciate you giving us the time.
Thank you.